Good morning, everybody. So today we are doing the second topic of plant biotechnology, that is somatic embryogenesis. I just want to revise you. Yesterday we did what is biotechnology. It is the just manipulation using the bioorganisms. Carl Reicke gives its name. Father is G. Haberinen of tissue culture. And these are the institutes. And technique to raise the virus-free plants is meristem culture given by G. Morrell in Cymbidium, that is orchids. And we have today what we do is the somatic embryogenesis and the concept of synthetic seeds or artificial seeds or encapsulated seeds. It is given by T. Murashige. Right? So now I will start the today topic. That is somatic embryogenesis. Somatic embryogenesis is an powerful, valuable biotechnological tool for the rapid multiplication, germ plasm conservation, and genetic improvement of elite hybrids and cultivars. So, somatic embryogenesis means it is the raising of somatic embryos. In orchids, these somatic embryos are called protocombs when it raised from the seeds, and when from the leaf they are called protocomb like bodies. And these protocombs are bipolar. And somatic embryogenesis and vegetative propagation, where somatic cells can generate secondary embryos due to totopotency of plant retracting from the embryogenic pathway towards the de-differentiation pathway. I already told you the concept of cellular totopotency given by Morgan, and cellular totopotency is the capability or ability of a cell to form a complete plant, and it has two pathways, two processes, phases, that is differentiation and de-differentiation. Here somatic embryos, they are formed by the de-differentiation system. And somatic embryogenesis of two types. First is direct. When somatic embryos develop directly from the explant, we can take explant anything, leaf, root, node, or floral organs like sepals and petals, without callus formation, that is DSC, called direct somatic embryogenesis, results in pure line development, avoiding soma-chrono variations. Explant, we have protocomb like bodies. I told you from seed it is protocomb, and from other part, then seed, that is explant, leaf, root, other floral organs, stem node, it is protocomb like bodies, and they form complete plantlet. Here there is no callus phase, there is no soma-chrono variations, so the plantlet formed, they are of same genome, that is pure line is formed. This is cell line, we can say cell clone, right? So see, this is the direct. We can put explant as leaf. In MS medium we did yesterday, and it gives rise to somatic embryos, that is PLBs, and then plantlet, and this is called direct somatic embryogenesis, and there is no callus phase. And these plantlets, they are pure line, homozygous, and tested to the cytological level for its stability. And the second type of somatic embryogenesis is the indirect somatic embryogenesis. When the somatic embryos are punctuated, means intervened through callus, it is called indirect somatic embryogenesis, also called IDSC. And the question arises, what is callus? Callus is the unorganized mass of cells. This is. Second phase is that it form a callus, which is unorganized mass of cells. Medium is same, and then these callus give rise to bipolar somatic embryos or PLBs in the root. This is called indirect somatic embryogenesis, IDSC. But as a tissue culturist, no tissue culturist, and you also, not supposed to prefer this path. As a tissue culturist, one has to prefer this one, because here chances in this is basically no chance of soma-chrono variation, but here it is high, because in callus there is a break in spindle formation, and soma-chrono variations are formed, which is our next topic we are taking tomorrow. and after doing that topic I will move to the molecular biology. So now Kallenbach 1978 classified embryos in the following ways. Number 1, zygotic embryos. In zygotic embryogenesis, two haploid cells combines to form diploid cell, that is called zygote, that is seed culture we say. But there has a problem in tissue culture. In seed culture the heterozygosity will come, and tissue culture does not prefer this type, that is zygotic embryos, because there is a fusion of genes and it results in the variations. Second is the non-zygotic embryos. It is formed from cells other than zygote. It is a following. First is somatic, we did right now, which are raised from the explant, that is tissue, that is maybe leaf, root, node or floral organs. Maybe mainly induced from somatic cells, in vitro or in vivo, that is somatic embryogenesis occurring in nature in ovules of Peonia. This question will come. And second is the adventive embryos. Here the somatic embryos arise directly from other embryos or organs. Example, citrus is developed from nucellus cells. In carrot it develops from the callus cells, which is derived from the root and petioles. And the next is the parthenogenetic. That is, parthenogenesis is the form of autogamy in which embryo comes from an unfertilized egg. And last, which we do next topic when we come back from molecular biology, that is the anther and pollen culture, that is androgenic embryos, which are formed from the male gametophytes, anthers and pollen grains, and they are basically haploids. Very important through the genetic studies, because they have haploid genes, and these haploid genes, they have to express whether it is dominant or recessive. So now question is the factors affecting somatic embryogenesis. Explant and juvenility. Among the factors, type of explants, leaf, root, petiole, embryo play the important role in influencing the induction of somatic embryogenesis. And the second factor is juvenility. First is the explant, and juvenility, that is age of plants, because age of plants have different level of endogenous hormones, and besides that it has physiological age plays important role in regeneration due to plasticity of cells. The cells are more biochemically and physiologically active, and the high level of endogenous I already told you, and that is why they are more acceptable to regeneration, whereas the higher the old age or with explant from mature plants, they are recalcitrant to the regeneration. They can't regenerate despite the nutrient regime in the medium. And the second topic is the genotype. The genetic constitution plays significant role in regeneration due to the endogenous level of hormone cytokinin like alfalfa, cotton, maize, soybean and rice. The genotype causes variability in the response due to different genetic, cytoplasmic and nuclear genes and their interactions. So genotype is very important, because I did my PhD in orchids, and we see some orchids they respond to BAP and some to kinetin. So this specificity to the BAP or kinetin is because of the genetic variability. Now we are coming to the medium. I already told you yesterday there are two medium, natural and that is synthetic. But natural medium is not reproducible because of it has not set composition. So in medium, the organic supplement in the medium plays an important role in inducing somatic embryogenesis. That is, coconut water known to have cytokinin activity, beneficial for inducing regeneration. Cytokinins promote cell divisions and direct somatic embryogenesis. Whereas auxins, particularly 2,4-D, it results in the formation of callus. But I already told you, as a tissue culturist, you are not supposed to get that pathway. Callus should be omitted to avoid the somocron variations. So the other factor we came to light, an external and physiological factor. It regulates the in vitro regeneration. The incubation of cultures under continuous light or darkness results in the suppressed somatic embryogenesis, that is, it is punctuated by the callus. The low light intensity favors rooting in date palm, whereas in orchids it favors indirect somatic embryogenesis due to the degradation of endogenous IAA. Hence, it is concluded that light requirement, like I told you, growth regulators, is species specific. It is vary with the genotypes. So now I am coming to the application. Before coming to application, I just want to revise that somatic embryogenesis, it is the raising of somatic embryos from the explants other than the seed. It is two types, direct and indirect. Indirect, we can get direct plantlet through PLBs or somatic embryos. Is no callus phase, so there are pure lines, homozygous, whereas in indirect it is punctuated by callus, which is the callus which is called unorganized mass of cells, and it is basically results in the somaclonal variations, because callus, the spindle breaks down and somaclonal variations, so somaclones formation are more prominently seen in the research. Now I am coming to the next, that is the application of somatic embryogenesis. The most promising application of somatic embryogenesis is rapid clonal propagation. I will show you this. If this is PLB, you can break into 4, right, and you put in the 4 tubes, and each tube you have 40, so you can break again into 4, so it becomes 160. So 160 each break into 4, and you have 640 again into 4. So like that, in this is first cycle, this is second, third. So in 5th cycle you have lakhs of PLBs, and these PLBs, they are then underwent acclimatization and forms the plantlet, and we can get approximately in 7 to 8 months the 1 lakh plants, whereas in nature it will take 7 to 8 years. So it is the very rapid of multiplication of economically important plants, and basic important is that in nature there is a possibility of heterozygity, because here we get the pure line clones. Production of artificial seeds, and efficient and low cost delivery and efficient system for germplasm conservation. So this is very important, because formation of this artificial seeds is important, as it is a low cost delivery and efficient for the germplasm conservation, right. I want to show you, and while talking, see, this is the basic formation of artificial seeds we did in labs. I made these seeds in 10 orchids, and see, leaf we culture, and we get PLBs, and we then put that PLBs into the solution called sodium alginate, and this sodium alginate solution of here it is, but we take the 3%. So 3 gram in 100 ml of medium, that is mithritol, and then we put that solution of, make a solution of calcium chloride. Calcium chloride is basically solution made of 100 millimolar, and these then we take with the pipette the PLB, suck it, and here we have the shaker and Teflon, and it start moving, and then we put drop wise these inside. As soon as these come inside, that is sodium alginate comes inside calcium chloride, so the beads are formed which are of calcium alginate beads and sodium chloride. After preparing, we can taste solution is salty. So these synthetic seeds are formed, in which this is PLB, which act as the embryo, and in outside this is endosperm, which is made up of medium, and outer layer is the testa, which is calcium alginate. So as these are synthetic, artificial, but they are like seeds, so they are called artificial seeds, right? So these are the basic structure, and they form like this. See, they look like this, and if I want to take to Delhi 1 lakh plants, I can take in very easily in 5 to 6 flasks, whereas in other way I have to hire a truck, and it will, lot of cost is there, and it makes very difficult to manage these things. So hence the production of artificial seeds and efficient and low cost delivery and efficient system of germplasm conservation. As I told you, it shortens breeding cycle, because here I will take only 7 to 8 generations, whereas in nature I have to take 8 years. Somatic embryos are best suited for direct gene transfer, which we do in subsequent days, that is direct gene transfer formation of transgenic plants, and somatic embryos are best suited in production of these transgenic plants. What are these transgenic plants? A very simple definition I myself gone through, that is when the donor is prokaryote and the receiver is eukaryote, the plant form is transgenic, right? But we did in detail in coming days. So the greatest importance of somatic embryogenesis is its practical application in large scale vegetative propagation. So vegetative propagation means there is no sexual phase involved. That is why it is shorten the breeding cycle. And third, the plants which are raised are basically pure line in culture, which I told you in direct somatic embryogenesis, right? So this is the concept of direct embryogenesis. So next topic I am doing the somoclonal variations, but if you take this phase, so there are chances of I already told you spindle breakdown, somoclonal variations, that is formation of somoclones, which is our next topic. So but it is not the somoclonal variation are bad, but there are 2% of somoclonal variations or somoclones. They are very important, and they help us in the improvement of crops, against disease resistance, against yield. So somoclones are good also. So there are two theories of somoclonal variation I will do tomorrow. So before that you have to do the somatic embryogenesis in detail, because somatic embryogenesis is a small topic, but it is a very important topic, right? And this diagram is its basically the life of this topic. And besides that, we have the these factors. Here I want to tell you again, please, today you have to after this lecture, so you have to do one thing, 5 macro you have to revise. So I told you the way of revising that macros, very easy, right? 170, 370, total is 540, but जोर का झटका लगेगा 440, 1650 and then 1900, right? This we know: potassium, ammonium, calcium chloride, magnesium sulfate and potassium dihydrogen. Then we have the micros. First 0.025 calcium, copper sulfate and cobalt chloride. Then I have 0.25, right? This total with 0.75. Then we have 0.83 potassium iodide. Then after 0.83 I have next is the point I will put, and last is it we have the vitamins. And vitamins we have pyridoxine HCl, thiamine HCl, inositol 100, glycine 2. So these are the very easy. And carbohydrate is sucrose 20 grams, and gelling agent is agar agar, that is 9 gram, which is obtained from the red algae, that is the Gelidium and Gracilaria. So this is the media we did. I already told you here media is MS. So today you will revise MS medium and somatic embryogenesis together, and tomorrow I will start the Somocron variations and finish it out, and then I move to the molecular biology. So please do this, and I assure you it is easy for you to clear the UGC, because these are the topics which basically decide the fate in the UGC. 90% questions they comes from these topic, right? So stay indoors, stay safe, take care, bye bye.